Yeah, cool, perfect. So yes, my talk is, as you mentioned, brain-wide visual habituation networks. So we did this work in larval zebrafish, and as Julian mentioned, the main advantage of larval zebrafish is that because they're very small and transparent, we can use different optical tools to see what is happening in their brains. And as I will show you in this talk, we exploited that to look at the whole brain calcium activity using GCAMP. And we saw with that tool, we could see how habituation learning was changing through multiple stimulus presentation. So I will first quickly mention what habituation is. Habituation is a type of non-associative learning, which is affected in some psychiatric conditions, and therefore that's why there's some interest on studying it. And it's, for example, in schizophrenia or autism, sometimes habituation defects are found. And it basically is a reduction in the response to a stimulus after multiple stimulus presentations. And there's a few characteristics of habituation and are like signature characteristics. And one of them is that after a small period of rest, when there's no stimuli presented, once you again present the stimulus again, there will be a recovery and then further habituation with more presentations. So we wanted to test if larval zebrafish will habituate to a very salient stimulus, a visual loom, which is simulating an approaching predator. So it's an expanding object. And other labs and work in our lab have shown how zebrafish larvae, when they see this stimulus, they will try to escape. I hope you can see the video on the little zebrafish here trying to escape from it. So we did some behavioral experiments, and we recorded the fish swimming, and we could flag the swims that were fast enough to be considered escape responses. And then we presented, we could see how this would change after multiple presentations. So our stimulus train consisted in 30 looms divided in blocks of 10 with 5 minutes, 2 5-minute periods of rest. And what we saw is that the probability of escape responses will decrease as we increase the number, well, the further presentations were coming on. And we also saw the characteristic recovery signature of the habituation learning. So we were very happy to see this. But this was also not very impressive, I must confess, because habituation has been shown in pretty much all animals. We were kind of expecting having these results. There was to test that it will work. But what we really wanted to see was what was happening in the brain. And again, because of zebrafish, we are able to do this. So we use a light sheet microscope that is with a system of mirrors allowed us to scan the brain very quickly. So we embed the larvae inaugurals, we mobilize it, and we put them in these little chambers or little aquariums on which then we can image them through calcium imaging. So here is an example of one of these scans going from ventral to dorsal. So the fish is looking up, it's looking that way, and all these little bright and gray circles are individual nuclei of neurons that will change in fluorescence depending on their activity. So I present my stimulus with a screen from on the right side, and as you can see in this example movie, the contralateral optic dectum, which is equivalent to the supercolliculus of mammals, here has a change in fluorescence when this animal was seeing the looming object. And there's also some other parts of the brain where you can see some changes in fluorescence. So this is the kind of data sets we're working with. And so this is on one plane, but we have up to 50 planes. But it's almost the whole brain. And to analyze the data, we have a workflow on which we first use software called Cayman, developed by another group, that allows to segment and detect the individual RIs that usually correspond to individual neurons. Then we use some clustering methods to find similar type of responses and the ones that are responding to our stimulus strain, the one that could be interesting for us. Then we use a linear regression to filter some of the responses that are not like clean enough. And then because all these RIs have 3D coordinates of each individual fish, we can warp them into a zebrafish brain atlas and that is also available and that was made available by another group. So in this way, we can find the different types of responses to our stimulus strain, and also where in the brain they are located. So these are the four main kind of responses that we found. So we had a big number of neurons, very widespread across all the brain, that are strongly habituating, so they habituate very fast. We have a group of moderately habituating neurons that habituate gradually, are more sensitive to the recovery, and they never fully habituate, at least not after 30 looms, and they're mostly located in the optic dectum. And then we have a group of weakly habituating neurons that habituate just a little bit, and they're more or less stable in their responses, and we can find them in the thalamus, the venula, and the optic dectum too. And then we have a group of inhibited neurons that seem to decrease their activity once the stimulus is presented, and we found them mostly in the rostral thalamus. So these were just some of the examples of the places where we found some of these responses. And so once this paradigm of this protocol was working and we were happy with it, we also decided to test it in several fish model of the fragile leg syndrome. So the fragile leg syndrome is caused by a single gene mutation, the FMR1 gene. This gene encodes the FMR protein that is involved in synaptic plasticity. And people with this syndrome usually have social, cognitive, and sensory alterations, but they often also have habituation deficits. And because the protein is highly conserved across taxa, there was a group 10 years ago now that was able to make a several fish model of it. So we first tested in our behavioral setup is the mutants will have habituation deficits, and we saw mild but clear trends on the fish here in red, the mutant fish, having slower habituation rates compared to wild types in blue, but also slightly stronger recovery. So this was encouraging, and then we went further up to using our light sheet microscopes to see what was happening in the brain, and to be able to compare across different fish and different groups of the fish, but also to see how the different parts or the different neurons or parts of the brain were interacting with one another, and also the different types of responses were interacting with one another. We decided to do network analysis. So to do so, we kind of subsample all the outriders, the thousands of outriders that we found that were responding to our stimulus strains into a few nodes. And these nodes will recruit the outriders that were in their vicinity, so close by, and also depending on the type of response they belong. And then we could do cross-correlation at each of the stimulus presentations. So let's say at each of these spikes that we see, and see and compare the level of correlation between all the nodes at different time points. So for example, this is for the first loom. So we see a lot of high correlation values because of course many nodes will have responded. But then at the 10th loom, as the network is habituating, we only see higher correlation values in the nodes that belong to the weakly habituating cluster. So using these correlation matrices, we can also generate networks that we could then analyze in many ways with, there's a lot of tools and very interesting things that we can look with in graph theory. And I won't have the time to present some of these results. I will be happy to mention it later, or if somebody is interested, we can talk afterwards. But I will just mention that we managed to find some differences between the two genotypes in terms of the rate of habituation of the network that were similar to the behavioral phenotype that we found. So for example, here in the wild types in blue, when we ordered the nodes based on their type of response, we see that for the second and third loom and then up to the 10th, the connectivity, the connections across nodes is decreasing, and then at recovery with this, some of them get re-engaged. And for the mutants, it also happens. There's also an habituation, but it's slower, and there's more connections that prevail as the habituation is occurring. And at the recovery point, there's also more connections that get re-engaged compared to the wild types. So yeah, in summary, several fused larvae habituate to looms, and we can locate the neurons that habituate to the looms and their different habituating profiles. And FMR1 mutants shows lower habituation in both behavior and network dynamics. And with that, I would like to wrap up. And I would like to thank all the people in the lab. It's a really nice lab, and we help each other a lot. And also to thank all the collaborators. And thank you very much, also you, for listening.
Thank you very much, Emmanuel. That is a powerful demonstration of the potential for the fish to give up its neural network's secrets, and remarkable that the FMR1 gene has these functions and impact on animal behaviors that can be sensed in this really powerful way. And so can I please start with the question with regards to FMR1 gene disruption in these lines of fish. And I might have missed this, but is their network organization different? And so are there quantifiable differences in the neuronal nuclei in these connections that you're measuring within these looms? And so are there any general anatomical differences that one should consider when we're looking at to understand what the differences in the networks might be?
Yes, and that's a very good question. So we did analyze the number of AURIs that we were detecting, and so the detection of AURIs have different steps, and the software allows us to detect pixels that have different activity changes, and that's how it segments into different neurons. And it detected the same amount of AURIs, or more or less the same amount of AURIs between wild types and FMR mutants. However, we usually detected less AURIs that pass or filters for AURIs that were responding to our stimulus strain. We don't know exactly if that's because maybe some of the neurons will be, because maybe they're highly correlated, some of them will be responding very similarly, and therefore the algorithm could be merging some of them, or if it's just because there's less AURIs or less neurons being sensitive to the stimulus strain. We haven't been able to completely find that out. But still, even though we saw differences and there were trends of it, we did analysis and they were not significant. So it's something that we have to keep on following up on it. And then on the network, the differences, so we analyze them in different ways, and there's, as I mentioned, different ways in which you can, and I will try maybe to see if I can show some of them very quickly. But for example, the participation level, this is a way to analyze the activity of the network. Participation is with how many different clusters or different identity of clusters that each node communicates. So if it's always, for example, a green node always communicating with green nodes, then it will have a low participation. If it's a node that communicates with red, green, blue, purple, then it will have a high participation. So one of the things we found is that the FMR1 network, oh, sorry, that's not in the FMR1. The FMR1 is a bit forward. Oh, yeah, here. They prevail with higher participation. So it means that it's not only more connected, but it's also more like kind of dispersing these connections, and also the recovery, this happens also more. So it seems that their network is a bit less stable. The way in which habituation occurs and at the recovery point, it follows a similar trend, but the connectivity will be kind of more unstable and like promiscuous, if you want to say so.
Right. That's yeah, really informative. Thank you. For the interests of time, and I understand it's quite late for some of us in the Northern Hemisphere, if I may, I should like to now hand over the floor to Jerry. And Jerry, can I ask you and all of our participants in this session to activate your videos if we can all be present on video, And Jerry, can I ask you to take everyone on a 3 to 5 minute overall discussion of the sessions while our judges deliberate on the prizes, and I'll get back to you, Jerry, with a person-to-person chat to tell you when we are coming back online. Thank you very much for that. Yep, great.
No worries. Have fun. Thanks everybody for the great talks. I really enjoyed them. I thought the range of research there was excellent, and on behalf of BioLegend, I'm very happy to sponsor the session. Does anyone have any pressing questions? I'm kind of put on the spot here, so I don't particularly have any discussion points apart from specific questions for speakers. But if anyone has any general questions, please let me know.
One from Emmanuel from my question was like, so at the beginning of your talk you were showing that there was 20 or 30,000 like different neurons that respond to the loom, right? And then when you were later in the talk where you were wanting to compare different fish to each other, you had to kind of reduce that to like 30 nodes, and you could still determine specific patterns and stuff. So I was wondering, it's more of a philosophical question, but like, why does the zebrafish have like 20,000 different neurons that respond to a loom, where you get enough information from just like a much smaller number when you did the analysis? Like, so is it just a lot of redundancy built in, or like why is it so?
Well, I guess it depends. I mean, I labeled the neuron space mostly because we were focused on habituation as like strongly habituating, moderately habituating, and weakly habituating. But we have to remember also that these neurons are also probably doing other things. And for example, the strongly habituating neurons, and why there's so many of them, we think they're mostly being part of a network of the fish being startled or scared. Like some of the areas where these neurons are located, they're related to fear responses to premotor areas and things like that. So there's obviously some redundancy, but I think there's also they're playing different roles and in different parts of the brain. For example, the weakly habituating neurons, some of them we have done further experiments and that I didn't have time to show, but some of them are actually just sensitive to the drop in luminance, so like dim sensors. So because of the narrative of the study, we focus on the habituation. That's how we were describing them. But again, we have to think that these are part of a whole system that is doing multiple things at the same time. And the reduction, I guess, was useful for us because it allowed us to do some of the analysis, because again, there will be different number of neurons in, and the locations will be slightly different across individual fish. And this method allowed us to do like this comparison across fish and across groups. But I guess, I mean, I felt kind of bittersweet about it, because also one of the nicest things about these animal models is like the amount of detail you can get, right? Almost single cell labeling activity. And we were kind of losing a little bit of that variance, which I think a lot of it can be very interesting for individual fish. So maybe we can one day go back into this data set and try to look for some other interesting questions at more of a smaller level, and see what are all these neurons doing, kind of going back to your question, like what are the roles?
Hello everyone, my name is Jerry and I'm one of the technical application scientists for BioLegend in the Australia and New Zealand region. I'd firstly like to thank the ADNF committee for organising this event and giving BioLegend the opportunity to sponsor. I think this is a wonderful initiative and I'm very much looking forward to all the talks today. So BioLegend is a privately owned global biotech company incorporated in 2002. We're headquartered in San Diego, California, and this is a picture of our new headquarters, which opened in mid-2019, along with a picture of our founder and CEO, Jean Lay. Our mission is to enable legendary discovery from research to cure, and we aim to fulfill this mission by developing technologies and manufacturing products across research areas, including but not limited to neuroscience, stem cells, immunology, proteogenomics, and oncology. I'd also like to mention that BioLegend is always interested in forming partnerships with researchers. As an example, we work actively with groups at the Garvan Institutes and also other institutes around the world to test emerging technologies. So if there are any technologies that you'd like to work with us on, or if you would like to discuss anything I talked about today in more detail, please get in touch with me or my colleague Hongyan, or you can check out our website. I've also listed the contact details of our distributors in Australia and New Zealand, and I will stay around in the chat, so please feel free to message me there. Thank you very much.